Yo, what's up, guys? I know we've already talked about it a bit, but I wanted you to have some formal notes on the structure of DNA. So then when we get to test time in a couple days, you'll just have a really easy resource to use. It's just straight to the point that is going to be in the same format as the test questions. So I'm going to show you a few things, and you're just going to fill in the note sheet as I go through these. All right, so we already know DNA, it's a nucleic acid. The sugar is deoxyribose. So it stands for deoxyribonucleic acid. Simple. Okay, and then tomorrow we'll start talking about RNA and look at the similarities and differences with that. All right, so remember, Watson and Crick get all the credit for discovering the shape, the double helix of it, but really it was Rosalind Franklin. She should have got the credit. She was the rad scientist that actually did the dirty work. All right, so yeah, Rosalind Franklin should get credit. All right. So in terms of what it's used for, why it's important, that's where we're going with the next few days. All right, let me. Okay, sorry. So in the next couple of days, we're getting into protein synthesis. So how is it actually used? It's back on, sorry. So how is it actually used? How does your cell actually make proteins using DNA? How does it actually make your traits? That's where we're getting into, all right? And we actually didn't even know what DNA was until pretty recently, like 1950s. That's when the structure of it was discovered. We didn't really understand what genes were. We didn't understand how they worked. Most of the field of genetics is like a new field that's growing really fast. Okay. So it allows us to understand medicine, health, how to genetically engineer things like crops. Like understanding how DNA worked is just opened a whole field of possibilities for us. A whole world of possibilities. Okay, so the shape, we already know it's a double helix. You might want to call it a spiral staircase, twisted ladder, whatever you want to call it. Two spiral strands. DNA is two strands, two sides, connected with those bases going between them. Remember? Okay, so remember the monomer of nucleic acids is called nucleotides. So DNA is made of chunks called nucleotides. These little monomers or subunits called nucleotides. And remember, each nucleotide has those three parts. Remember? They have a phosphate group, a sugar, and a nitrogen base. So the base, that's the only difference. There's an A base, a T, a C, and a G. Adenosine, tyrosine, guanine, cytosine. That's the only difference between the different nucleotides is the base. The sugar and phosphate is always the same. Okay, so those three things together make one nucleotide. Okay, so the sugar in DNA is deoxyribose. Sometimes I'll just say sugar, but even when I say sugar, that's what I mean, deoxyribose. Okay, and the four bases, like I said, adenine, cytosine, thymine, and guanine. Usually we just like to save time and say the first letter, A, C, T, or G. Okay, the T and the C, these are what we call pyrimidines, which means if you look at the shape of the base, it only has one ring of carbons. And then the A and the G have two rings. So they're called purines. Okay, I don't think I'll test you on that. You don't need to know which ones are purines and which ones are pyrimidines. But this is why the A and the T go together and the C and the G go together. Because one has one and one has two. And you want to have a total of three. All right. Okay, next step. Remember the sides of the ladder, like how I drew it, it just goes sugar, phosphate, phosphate over and over and over and over. That's it. All DNA on the whole planet does that. Sugar, phosphate, that's it. It's always the same. Okay? And then connecting the two sides are the rungs of the ladder. That's where you have the bases, where T's and A's are always together, and C's and G's are always together. You always have two of them connected, like puzzle pieces. And remember, those bases are held together by hydrogen bonds. And those hydrogen bonds are weak. And they have to be weak because you're always unzipping your DNA and using it. So the only way you can unzip it and use it is if those are weak bonds that you can break really easily. Okay, so you saw yesterday the Chargaff rule. Basically, we know that A's and T's go together because if you look at the DNA and count how many A's there are, it's always the same as the T's. And if you count how many C's there are, it's always the same as the G's. So the guy that discovered that, his name was Chargaff. So we call it Chargaff's rule, that the A's and T's are always equal to each other. And C's and G's are always equal to each other. Okay, so this is also called the base pairing rule. The A's and T's are always paired together. C's and G's are always paired together. Sometimes we call this complementary. If I said, yo, Jane, your outfit is dope today, that would be a compliment, right? With an I. This is complement with an E. Complementary means that two things go together, like peanut butter and jelly, like they need each other. So base pairs are complementary. T is complementary to A. C is complementary to G. It just means they go together. Complementary. Okay? So if I said this top strand, the AT, if I said that's one side of your DNA, you know the other side of your DNA. Because you know that T would go with the A, and the A would go with the T, and the G would go with the C. So if I give you one strand of DNA, you should be able to tell me what is the second strand, what is attached to it. Okay, and that's pretty much it. All right. So after you do these notes, after you get all these notes filled in, there's a little website you're going to go to. It's like Nobel Prize dot something. You're going to go to this. It's a simulator where you're actually going to build a strand of DNA and then just like record your results and show me a screencast or a print screen or something after you're done, just to prove to me that you did it. Show me your results. All right. And that's pretty much it for today. Okay, and then you're going to do a Kahoot. There is a Kahoot challenge that in Google Classroom, you just click on the link. You might need a PIN code, and I put the PIN code in the instructions. So after this, go do the Kahoot challenge, and you guys are good for the day after that. And I'll see you tomorrow. All right, see if I can get this thing turned off here.